Good day, everyone. I am Maria Ibon N. Resano, a Dial Gensei student of Iloilo State College of Fisheries. This presentation is a course requirement for Biology of Microorganisms under Dr. Brian Ives Araneta. I am tasked to present an article entitled Evolutionary Changes of an Intestinal Lactobacillus reuteri During Probiotic Manufacture by Lazaro Lopez and his team. The International Scientific Association for Probiotics and Prebiotics describes probiotics as live microorganisms that, when administered in adequate amounts, confer a benefit to the health of the host. Probiotics are among the most consumed dietary supplements. In the USA, around 3.9 million people consume prebiotic or probiotic supplements. There are many questions about the efficacy of these products in the medical field. One of the crucial uncertainties is how long the probiotic bacteria can linger in the intestine. Bacterial adaptation to new environmental conditions is associated with the loss of adaptation to the original one. This adaptation occurs through the selection of mutants displaying greater proficiency to thrive in new substrates, temperatures, pH values, etc. Lactobacillus reuteri is especially relevant as a probiotic bacterium because it has co-evolved with different animal hosts, including humans, and secretes antimicrobial compounds that inhibit other bacteria. The researchers studied the evolutionary changes that occurred in an intestinal isolate of L. reuteri during 150 passages in milk. Isolation and selection of Lactobacillus reuteri strains. The strains used in this study were isolated from the duodenum of a female CD1 mouse. All procedures with mice were previously approved by the Ethics Committee on the Use of Animals in Research and Teaching at the San Francisco University of Quito. A CD1 mouse was euthanized using chloroform. The duodenum was extracted. Contents were inoculated into man-rugosa sharp MRS agar and incubated at 37 degrees Celsius for 48 hours under microaerophilic conditions. Isolation and selection of Lactobacillus reuteri strains. Isolates were analyzed by gram stain, catalase, and oxidase test in Appendix 1, Table A1. 6 Lactobacillus SPP isolates were selected, and 16S rRNA gene sequence was used to determine bacterial species. PCR products were sequenced at Functional Biosciences Incorporation, Madison, WI, and sequences were compared to those in GenBank. Selection of rifampicin-resistant mutants. Rifampicin mutants were selected following the protocols previously described. One L. reuteri strain was inoculated in 10 ml of MRS broth at 37 degrees Celsius under microaerophilic conditions for 24 hours. The bacterial culture received an additional 10 ml of MRS broth with rifampicin, 200 micrograms per ml, for a final concentration of 100 micrograms per ml. And the tube was incubated for another 24 hours. The bacterial culture was streaked on MRS agar with 100 micrograms per ml of rifampicin and incubated for 24 hours. Colonies formed by rifampicin-resistant mutants were confirmed with gram stain, catalase, oxidase, and antibiotic sensitivity test. Confirmed strains were stored at -80 degrees Celsius in BHI medium plus glycerol 20%. Fitness loss due to the mutation causing rifampicin resistance was evaluated by culturing together progenitor strains and rifampicin-resistant descendants as previously described. Adaptation to milk and MRS. Two lineages of L. reuteri were created by inoculating 1 milliliter of LRRR1.2 culture in MRS broth in 2 tubes containing 9 ml of sterile, autoclaved cow's milk and incubated at 37 degrees Celsius for 24 hours. Each of the lineages was subjected to 150 serial passes in milk, which corresponds to approximately 510 generations. The strain adapted to MRS was obtained by 150 passes in this medium, and the same procedures used for milk. To rule out potential lactobacillus contamination, PCR amplification and sequencing of LEO-S gene was carried out at passes 55, 92, and 120 in each population. Intestinal colonization. Male and female CD1 mice were donated by Laboratorios Agrocalidad Quito. Animals were fed for 7 days with 100 microliters of MRS medium containing 2 × 10⁷ L. reuteri cells, as determined by Petrohauser chamber count and confirmed by colony count and MRS plus rifampicin. The animals were separated into 3 groups. Group 1 was fed with LRR1.2 non-adapted to milk. Group 2 was fed with LRR1.21 M150 strain better adapted to milk. And the Group 3 or control group was fed with 100 microliters of sterile MRS medium. The mice received the inocula once a day for 7 days and left untreated for 15 days to ensure colonization, after which they were euthanized. A 10 mm duodenum piece from each mouse was ground with MRS medium, and the resulting menstruum was diluted serially, 10-fold, plated in MRS plus rifampicin, and rifampicin-resistant colonies were counted after 24-hour incubation. Relative fitness in milk. The researchers compared the growth rate in milk of the adapted strains LRRR1.21 M150, LRRR1.22 M150, and LRRR1.2 MRS150 with that of the parental strain LRRR1.2. They inoculated 1 ml of a 24-hour culture MRS of each of the strains. LRRR1.21 M150, LRRR1.22 M150, LRRR1.2 MRS150, and LRRR1.2 into 5 tubes containing sterile milk, 5 replicates per strain, and colony counts were obtained at 0 hour and 24 hour of incubation. They used the spread plate method in duplicate. The relative fitness was calculated using a previous described formula. W is equal to Ln times A 24 hour over A 0 hour over Ln times B 24 hour over B 0 hour, where W is the relative fitness. A24 corresponded to the colony count of each of the adapted strains. LRRR1.21 M150, LRRR1.22 M150, and LRRR1.2 MRS150 at 24 hour of culture. A0 corresponded to the colony count of each of these strains at 0 hour of culture. B24 hour corresponded to the average colony count of the 5 of the ancestral strain after 24-hour incubation, and B0 hour was the colony count average of the 5 replicates at 0 hour. DNA extraction and whole genome sequencing. DNA was extracted from the three strains LR1, LRRR1.2, and LRRR1.21M150, the strain showing greater adaptation to milk, using DNA-Zol-TM in vitro gene, following the manufacturer's protocol, and sent to the Microgen Incorporation Seoul, the Republic of Korea, for genome sequencing with Illumina HiSeq 2500 100 BPPE. The complete genome sequences of the three strains were assembled using SP8's version 3.13.0. The raw data of the sequences were pre-processed before running the hashing. The researchers used paired-end reads from SP8's using MOV version 2.4.0 for the reordering of the contigs. Based on the complete genome sequence of L-Reuteri DSM 20016 on GenBank. Fitness or relative fitness in milk. Both milk-adapted strains outperformed the ancestral one. However, only one strain showed a relative fitness that was statistically significant, values above 1. LRR1.21M150 showed a W equals to 2.92, 99% CI, 2.62 to 3.22, whereas the strain LRR1.2 22 M150 had W equal to 1.35, 95% CI, 0.99 to 1.71. The relative fitness of the strain adapted to MRS medium, LRR1.2 MRS150, was negative, W equals 0.98, 95% CI, 0.63 to 1.33. Intestinal colonization. The duodenal samples of mice receiving different strains of L-Reuteri were subjected to bacterial culture, but the researchers obtained colonies of L-Reuteri from two mice only, one from the mouse receiving the parental strains and the other from a mouse receiving the milk-adapted strains. Nevertheless, the mouse receiving the parental strain LRR1.2 had 35 times higher colony counts than the animal receiving the milk-adapted strain LRR1.21M150. 993 colony-forming unit per ml versus 28 colony-forming unit per ml. Whole genome sequencing. The alignment made in MOV showed that all the contigs were correctly aligned among the three analyzed genomes. Comprehensive genome analysis in Patrick showed that the three genomes have 2,320 MB, 2,303 MB, and 2,296 MB, respectively, distributed in 310, 264, and 315 contigs. Genes annotation allowed us to compare 1,618 proteins with functional assignments and 647 hypothetical proteins from LRR1.2 milk-adapted strain with the LRR1.2 strain. Of the coding sequence genes analyzed, 6% had mutation substitutions, insertions, or deletions compared to the progenitor. Of the 13 mutations found, 8 were non-synonymous. 5 of them were in genes associated with metabolism, YHDG, an amino acid permease, YBIV, a sugar phosphatase, MURG, andecaprinol and acetylglucosamine transferase, AMT, an ammonium transport protein, and PER, a peroxide responsive repressor. Figure 1 showed relative fitness in milk of Lactobacillus reuteri strain isolated from the murine intestine, subjected to 150 passes in milk, LR 1.21 M150 and LR 1.22 M150, and subjected 150 passes in MRS medium, LR 1.2 M150 MRS. The bacterial growth was compared to the parental strain LR 1.2, 1 pass in MRS. Bars indicate standard error. The researcher showed that an intestinal L. reuteri isolate subjected to 150 passes in milk gained proficiency to grow in this substrate. They also showed some evidence that the adaptation to milk reduced the aptitude to colonize murine intestines. Their results are in agreement with previous studies showing that adaptation to a new ecosystem reduces the fitness in the original niche. A phenomenon is known as antagonistic pleiotropy. This finding is also in agreement with previous reports indicating that commercial probiotic bacteria originated from intestines failed to colonize mammalian intestines. A total of 13 mutations were observed in bacteria subjected to milk passes. Some of these mutations occurred in genes associated with metabolic processes, which have been observed in bacteria forced to grow in challenging environments. The gene coding for the permase YHDG, an exporter for branched-chain amino acids, showed a non-synonymous mutation. Mutations in gene have been observed in an acid-tolerant mutant of Streptococcus mutans. Lactobacilli growing in milk have to endure high concentrations of autogenous organic acids or lactic acid, which can be deleterious. Acid-resistant mutants occur frequently in bacterial populations subjected to these types of acids. A mutation in the AMT gene, an insertion of 21 nucleotides, was observed in L. reuteri adapted to milk. The product of this gene is involved in ammonium transmembrane transporter activity. This gene is overexpressed in L. acidophilus adapted to milk. The YBIV gene codes for a sugar phosphatase involved in bacterial protection from cystoplasmic phosphosugar accumulation. This mutation may have some association with the high availability of sugars in milk. Peroxide-responsive repressor per-R gene has a deletion of 34 nucleotides in the strain adapted to milk. In this study, the researchers showed that few passages in an artificial substrate can select mutants that can grow better in this novel substrate but unable to compete with ancestral strains in the original niche. However, the results should be confirmed by a larger experiment using more replicates. The results suggest that more effective probiotics may be produced with low passage strains. Finally, bacterial antagonistic pleiotropy is a phenomenon that has serious implications that go beyond probiotics, such as antibiotic resistance and the evolution of environmental opportunistic pathogens. Thank you very much for listening.